Hi, welcome to AP Biology. I hope you can see me. All right, I'm Gail Pfizer. I'm from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. And I'm here to talk about and help prepare you for our May 18th, 2020 AP Biology exam that will be at 2:00 p.m. Eastern Standard Time. And there's going to be two questions that are two free response questions. And the first question is going to be about interpreting and evaluating experimental results. And you're going to have 25 minutes to read and respond, and then 5 minutes to upload. This is going to be 65% of your exam score. And this is going to be based on an authentic scenario accompanied by data in a table and or graph. And it's going to be built with 4 parts, okay? And there's going to be 1 to 2 task verbs in each of those parts. So let's talk about each one of those briefly. In A, you're going to describe and explain biological concepts, processes, models, and applied context. In B, you're going to identify and justify experimental design procedures. And C, we're going to analyze some data. And D, we're going to make and justify predictions. So let's talk about those task verbs really quickly. Describe, you're just listing the characteristics of whatever it is you're talking about. Explain is going to be the how or the why. Identify is going to be really short, just what is it? And then don't forget and justify. You got to make your claim. You got to make prove it with your evidence, and then support it with some reasoning, for some biological support there. All right, and then analyzing data, there is going to be a table or a graph that you're going to analyze. All right, in question 2, it's conceptual analysis, and that's going to be 15 minutes for that response. There's going to be 5 minutes to upload, and that's going to be 35% of your score. Excuse me. It's going to be based on an authentic scenario describing a biological phenomenon with a disruption, and there's going to have 4 parts. There's going to be 1 task verb in each of those parts. The first one's going to be describe biological concepts or processes in applied context. B is going to be explain biological concepts or processes in applied context. C is predict the causes or effects of a change in the biological system. And D is going to be support or justify a claim with reasoning and or there's that evidence again. So let me talk about our lesson for today. We are going to be just talking about, it's going to be very narrowed focus. We're just going to be talking about proteins and nucleic acids, and there's going to be a lot of work with the science practices built in. Excuse me. All right, so here's our warm-up. And our warm-up is from our homework from yesterday, and which was from, if you're just joining us now, it was from our previous lesson, which had to do with properties of water, what else? Monomers and polymers, and then carbohydrates and lipids. So here's our question that they had for homework. And if you're just joining us for the first time, then we're going to go ahead and give you a moment between the big reveal of the answer of this question. Okay. Carbon is a very important element in living systems. The following structures are examples of 2 different categories of macromolecules. Identify each category and describe how each type of macromolecule is important to the structure and function of living things. So you see in category 1, and I think that you can see my cursor move. I am going to count. I'm going to tell you how I can tell what this is, okay? So right now, well, let me see. There it is. Okay, so there's, as far as carbons, here's 1, 2, 3, 4, 5, 6. Okay, so I know that there's 6 carbons. My H's, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. So 6 carbons, 12 hydrogens. And now for our oxygens, there's 1, 2, 3, 4, 5, 6. So there's 6 carbons, 12 hydrogens, 6 oxygens. That's a 1-to-1 ratio. I know that's a carbohydrate, okay, by looking at it. All right, and then category 2, I see 2 long chain fatty acids, and I see a phosphate group here. So I know I must be in the lipid category, and I must be in, this must be a phospholipid, right? All right, let's look at the scoring guideline. All right, so category 1 is a carbohydrate. It's a monosaccharide. And then you could be specific and put the word glucose in there. It functions to provide readily available energy to organisms and also functions as a monomer of polysaccharides. In category 2, it's a lipid. It's specifically a phospholipid, and it's amphipathic, which means it has a polar end and a nonpolar end. And then it also functions as a component of the cell membranes. So we have down at the bottom, those are in my classroom, and my kids built those this year. And this is a very popular assignment, so you may have them in your classroom also. This is a dehydration and with carbohydrates, dehydration synthesis, and then we have some triglycerides hanging over here on the cabinets. Okay, so here's what you need to know. So the learning objectives, this is what you need to be able to do, excuse me, to apply to both proteins and nucleic acids. So we need to, I'm so sorry for that noise, but we were afraid about the sound, and so I needed to keep my email open. All right, so describe the composition of macromolecules required by living organisms, which that's where you're going to talk about the elements of each, and then describe the properties of monomers and the types of bonds that connect the monomers in biological macromolecules, and that's where we're going to talk about dehydration synthesis and hydrolysis and covalent bonds, and then describe the properties of monomers and the types of bonds that connect the monomers in biological macromolecules, and this is where we're going to talk about the structure of a nucleotide, the primary structure of amino acids, and those R group properties. And then explain how it changed. See how I highlighted that in red? Where do you think this is going to fit? This is going to fit in 2C, which had to do with predicting a change, okay? I not only want you to learn well how to describe, explain, but also be ready to talk about what happens if you don't have it or what happens if you change it, okay? No matter what you're talking about. All right, so how a change in the subunits of a polymer may lead to changes in structure and function of the macromolecules. We are talking about in this section nucleic acids, directionality, the 5' to 3', how DNA is anti-parallel, A goes with T, C goes with G, all that kind of stuff. Amino acids, we'll talk about the carboxyl terminus and how that's the end that we add amino acids to develop a polypeptide. And then we'll talk about 4 levels of protein structure. And we're also going to end it with describing the structural similarities and differences between RNA and DNA. All right, so first we're going to talk about proteins. So these are the molecules that you need to build proteins. You need carbon, hydrogen, oxygen, nitrogen, and sulfur. And you'll find the sulfur. I have a diagram of cysteine right here. Cysteine and methionine have the sulfur included in their molecular structure. So here, let's go ahead and talk about how this is built. So right here, this is our central carbon, okay? And then it always has this hydrogen. And then this group right here, this is our R group or our R chain. And all of those will differ from amino acid to amino acid, and that's what gives them their different properties. On the right here, we have our amino functional group, okay? And on this side, we have our carboxyl functional group. All right. Okay, so let's do dehydration synthesis first, and then we'll do hydrolysis, and we'll do both of them with proteins, okay? So amino acids are monomers that bond together with a peptide bond, which is a covalent bond, to form a polypeptide, which is a polymer, through the process of dehydration synthesis. So let's look at, here's one amino acid, here's another amino acid. We're going to take the hydroxyl group and this H, and in the reaction, we're going to pull it out and we're going to make a molecule of water, okay? So now we have these 2 amino acids joined together with a covalent bond. You see this little gray arrow? That's where it happened, and that's called a peptide bond. All right, the process of hydrolysis is just the reverse. We're going to break apart the polypeptide into amino acids. So we're going to take this, there's 6 amino acids in this diagram. They have a reference to R1 through 6. And so we're going to take it, we're in a reaction with an enzyme, we're going to put water in, and we're going to bust it apart, and you see an OH and an H. There's the OH coming back on this carboxyl group and an H to this amine group. And we just took a 6 amino acid strand and made it into 2, 3 amino acid strands. Okay, so I talked about this, and this is the R group, remember? On this diagram, I circled them all. There's 1, 2, 3, 4, 5, 6 of them, and those are different in all of the amino acids. The R group of an amino acid can be categorized by its chemical properties: hydrophobic, hydrophilic, or ionic. The interactions of these R groups determine the structure and function of that region of the protein, and we'll talk about those functions a little bit as we move through this unit, but specifically when we get to their own unit in our units 1 through 6. Like we'll talk about cells and cells, and we'll talk about the proteins in cells, and we'll talk about the proteins in cell communication, and then the proteins and enzymes. All right, so I wanted to bring this up so you could understand why these R chains are the properties are so important. This is glutamic acid, and on the left, this one right here, and it's, see this R chain right here? It's very much different than that one, right? Well, this one has a hydrophilic property, and this one has a hydrophobic property. So if this one's hydrophilic, it means that it enjoys being on the outside of the protein and interacting with water, okay? And if you're hydrophobic, that means you don't want to be on the outside of the protein. You want to be on the inside of the protein, away from water. And so this is a big problem if you have one substituted for the other. And this is in particular in the case with sickle cell. And if you want to, I talked a lot about mutations, protein synthesis, and this substitution of these two amino acids in our natural selection video that was on March 26th. If you want to go back there for the whole story, there's a lot of good material in those videos that have to do with science practices and other material also. All right. So anyway, so sickle cell, you're supposed to have glutamic acid, and anyway, you end up with valine, and that's how you have the non-functioning cell. Okay. All right. So amino acids have directionality with an amino N and an H2 terminus and a carboxyl COOH terminus. And here is the amine. There's the H and H there, and here is a C, two O's in the H. This is a hydroxyl group. This is a carboxyl group altogether, okay? So we have if we're adding amino acids, they're going to be joined to this end, let's say, and they're also in that same video, there is also protein synthesis and transcription and how you read the genetic code from there, and all that's there. All right, and we'll talk about that again also in protein synthesis when that comes up again. All right, so we're going to add amino acids to this end when we are at the A and P site on the ribosome and adding amino acids to make a long polypeptide. All right, so proteins comprise linear chains of amino acids connected by the formation of covalent bonds at the carboxyl terminus of the growing peptide chain. All right, so we're going to talk about the 4 levels of protein structure now. The primary structure is determined by the sequence order of their constituent amino acids, and the specific order of amino acids in a polypeptide, which is the primary structure, determines the overall shape. So here, each one of these dots, okay, those represent amino acids. For example, I just threw these names on. Okay, there's arginine, there's valine, there's glutamic acid. Here's our carboxyl group. Here's our amine group. So this is where it begins, and this is where more amino acids are added to this end. That's our protein structure. It kind of looks like a pearl necklace, somewhat. All right, secondary structure. Secondary structures arise through local folding of the amino acid chain into elements such as alpha helices and beta sheets. So if you'll look at, here is see, this was that strand, but now it's in this alpha helical conformation. And so we have these black dots, they represent hydrogen bonds, and that's what's causing this to take this shape. Same with the beta pleated sheet. The dots there, that's representing hydrogen bonds, and that's causing it to form in this particular structure. All right, the tertiary structure. This is the overall 3-dimension shape of the protein, and it often minimizes free energy. It decreases the free energy to increase the stability. Okay, so we have hydrogen bonds, disulfide linkage. You might see that as disulfide bridge or disulfide bond, depending on what you're reading, and ionic bonds and hydrophobic interactions. So all of those are happening in this particular formation of this structure. All right, here's our quaternary structure. It arises from non-covalent interactions between the multiple polypeptide units. So all of those that we just talked about formed this tertiary structure, and then we have other interactions between this tertiary structure and this tertiary structure for the quaternary structure. And here they are all together. How this particular end result here will determine the function of it, okay? All right, so we have primary, secondary, and then tertiary, and then our quaternary. All right, I talked about this a moment ago. Some major functions of our proteins we'll discuss a little bit now and much later. Catalysis by enzymes, cell structure, and communication between cells. And now for our nucleic acids. Okay, so we'll go through it the same way we just did proteins. Okay, we'll first talk about the molecules that we need to build the nucleic acids. We need carbon, hydrogen, oxygen, nitrogen, and phosphorus. So here's in that phosphate um, um, phosphate group, that's where we're going to need the phosphate. So let's take a look. We can go ahead and do a little bit of breaking down. This is a nucleotide, okay? And this is a 5-carbon sugar. This is a nitrogenous base, and this is a phosphate group. And we'll talk about a little bit more in depth. Well, no, let's go back. Let's stay there for a minute. No, we'll go. So let's do I'll talk about the structure in a minute, but let's learn how to put them together first. Okay, so dehydration synthesis, we're going to form covalent bonds. We're going to take nucleotides, which are the monomers, and we're going to form a polymer, which is the nucleic acid, whether it's RNA or DNA. So here's our nucleotide. There's our phosphate group, our 5-carbon sugar, and our nitrogenous base. Now is where I need to talk to you a little bit more about the nucleotide itself. Okay, so if you're looking at this 5-carbon sugar, here's carbon 1, 2, 3, 4, and 5, okay? That's where the 5' to 3' direction language is coming from. It's talking about the orientation of the carbon on the 5-carbon sugar. So our phosphate end of one nucleotide, this one right here, this will be nucleotide 2, is going to bond with the hydroxyl, the 3' hydroxyl end of the previous nucleotide. So this H and that OH will go through a reaction to form a covalent bond. It's called a phosphodiester bond, and then we'll have those 2 nucleotides together. And then the same process of dehydration, whether we're talking about lipids or carbohydrates or amino acids joining together, that molecule of water is going to be formed, okay? So that's dehydration synthesis for nucleic acids. All right, so now let's talk about everything we can about this nucleotide. Okay, we see that it has a 5-carbon sugar, and I'm gonna walk through that again. There's carbon 1, 2, 3, 4, and 5, okay? The phosphate group, which is negatively charged, is on the 5' end, and then the 3' end, that's where we're going to be joining the next nucleotide, okay? All right, so what else did I want to talk about? The nitrogenous base. There's 2 types of nitrogenous bases. We have a purine and we have a pyrimidine. This is, there we go. This is my pyrimidine, and I know that is because it's a single ring. If it was double, then I'd know it's a purine. And my pyrimidines are my thymine, cytosine, and my uracil, okay? And then my purines are my adenine and my guanine. All right. So when we add a nucleotide to a growing strand, this would be DNA polymerase, probably, it's not labeled, but here we have a 5' end of this strand, 3' end of this, and then we have our complementary base pairs. We have our A's and our T's and our C's and our G's. So the next one would be, if this was an A, then that would be a T that's coming in. It's going to be joined to this 3' end, and a covalent bond will be made. And I'm going to go through the whole process of that nucleotide being added when we get to DNA replication, okay? All right, so it's called anti-parallel. So if you'll look, I tried to do an attempt of flipping some molecules around. So let's look at, here's a nucleotide, here's our phosphate group, here's our 5-carbon sugar, and here's our nitrogenous base. There's our carbons, 1, 2, 3, 4, 5. Now, if you, let's see if I can do this. I do this in my classroom. If you took it and flipped it upside down and around, so 2 turns, like up and down around, then you're going to end up with this orientation. And if you look, there's our 1, our 2, our 3rd carbon, 4, and 5. And everything is on the opposite side and turned and upside down, okay? That's called anti-parallel. And that is how the strands are oriented to form our double helix. And let's see. We talked about our 5' is on our phosphate end, 3' is on our hydroxyl end. Okay. All right, so now let's talk about the hydrogen bonds between the nucleotides. Cytosine nucleotides pair with guanine, and they have 3 hydrogen bonds between them. And then adenine and thymine, they have 2 hydrogen bonds between them. Remember that in a question that I'm going to ask you in just a little bit. Okay, so these are things similar between DNA and RNA. They both have 3 components. They have a 5-carbon sugar, they have a phosphate group, and a nitrogenous base, okay? They both have nucleotides that are joined together with a covalent bond. And they're both linear molecules, and they both have a 5' and a 3' end, and they both have nitrogenous base, which are perpendicular to the sugar phosphate, okay? All right, now the differences. RNA, here's RNA, single strand, DNA, double strand. 5-carbon sugar is called ribose in RNA because it's RNA is ribose, okay? And DNA has double strand, and that's deoxyribose. And then your DNA is going to have thymine, and then your RNA will have uracil and not thymine. And then DNA has also that anti-parallel directionality. All right, so that's it for the content. We'll apply it in some questions in just a little bit. Let's talk about what science skills we're going to do. We have 6EB. We're going to do a lot, but these are some specific ones that are unique to this presentation. All right, so 6EB, we're going to predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. And then 2B, we're going to explain the relationships between different characteristics of biological concepts, process, or models represented visually. So right now you should be thinking, oh, I see this in 1A and B and 2A and B on our test. That's what we're going to be doing, this explaining. And then the 6EB, that's going to fall nicely into that C of that question 2. All right, this is one of my favorite questions, and it's from probably the first AP exam since a revision that happened in 2012. Okay, so which of the following correctly illustrates a dipeptide and an amino acid in the optimal position to form a tripeptide? So let's take a look at A. All right, so here's our dipeptide, and there's our amino acid that we want to add to this, and the same situation all the way down. So let's start from the bottom. So we have an amine end here, and there's an amino functional group. This looks like a methyl functional group. Can we add those? Can we bond those together? Is that how we do this? No. And here we have an amino end and an amino end. Will that work? Nope. And then we have a carboxyl and a carboxyl. That's not going to work either. But look here, carboxyl and our amine end, and that will bond together. That's the way we add amino acid to the strand that we're creating, is to that carboxyl end. So A would be our answer. Okay, I know the bottom of this is hard to read, and I'll read it for you. Here we have this image that I keep using with the cysteine. So let me read this to you. Researchers compared similar proteins from related organisms in different habitats. They found that the proteins from organisms living in harsh environments had a greater number of cysteine amino acids than did proteins from organisms not living in harsh environments. The structure of cysteine is shown Bonds can form between the sulfur atoms of different cysteine amino acids, that SS bond, and that is a strong bond, a covalent bond. All right, so which of the following best describes the effect of a greater number of cysteine amino acids on the stability of the proteins? All right, so listen carefully to the choices. We have A, the change has no effect on the stability of the protein because only one type of amino acid is involved. B, the change leads to increased protein stability because of an increased number of SS bonds in the tertiary structure of the proteins. C, the change leads to a decreased protein stability because of an increased number of SS bonds in the tertiary structure of the proteins. And then D, the change leads to an increased protein stability only when the added cysteine amino acids are next to the other cysteine amino acids in the primary structure. Okay, so we're going to have increase. So we can go ahead, if I had a pen, I would be crossing out this decreased. Okay, nope, C is not it. A, no effect, that's not it. Now, do they have to be right next to each other in line? Like, did this have to join? Did another cysteine have to join right here for that? No. So the choice here that works for us, the change leads to increased protein stability because of an increased number of SS bonds in the tertiary structure of the proteins. Okay, so that's our answer B. All right. Now, I'm going to pause on these because I'm going to give you time to answer these. This is another one of my favorite questions. I always like to include this on everything that I give my students. They'll be doing this tomorrow. It was number 56 on I think of the first exam. Also, there were some really rich, good questions on that test. All right, so number 56, a culture of Spirogyra, an autotrophic alga, is maintained in a water solution containing dissolved carbon dioxide and a source of phosphates but lacking nitrogen compounds. That's key. A researcher determines the rates of synthesis of several organic compounds found in the Spirogyra before and after several weeks in the water solution. Which of the following graphs best illustrates a likely result of the experiment? Okay, I'm going to let you look at this for just a minute. I'm going to turn my timer on. I haven't done that yet. I'll give you 10 more seconds to think about it. Okay. All right, so let's look at this. What did I say happened? We are lacking what? Nitrogen. So what element, where is nitrogen? We're going to find it in proteins. We're going to find it in nucleic acids. So if we're not able to use nitrogen anymore, then the amount of proteins in nucleic acids are going to decline. So is it going to affect the carbohydrates and lipids? No, because they're carbon, hydrogen, oxygen, and then lipids had the phosphate for phospholipids. So let's look for one where everything's fine other than proteins and nucleic acids are declining. All right, so no decline here. And then we have a decline in proteins, a decline in nucleic acids. Everything else is holding steady. Okay, are we declining? No, we actually went up, impossible. And then we went up and we went down. So our answer here then is B. Okay, and then one more question from that same test. All right, so we're looking at two boxes. Here's segment 1, and we have, this is what I see. We have a strand of DNA, and in segment 2, we have a strand of DNA. They're in their normal 5' to 3', and they're anti-parallel, and they're complementary nucleotides. All that looks good. And then I see the sequence in segment 1 and the sequence in segment 2. Now the question, the sequences for the two short fragments of DNA are shown above. Which of the following is one way in which these two segments would differ? A, segment 1 would not code for messenger RNA because both strands have T, a base not found in RNA. B, segment 1 would be more soluble in water than segment 2 because it has more phosphate groups. C, segment 1 would become denatured at a lower temperature than would segment 2 because A to T base pairs have 2 hydrogen bonds, whereas G to C base pairs have 3. And D, segment 1 must be from a prokaryote because it has predominantly A to T base pairs. So take about 10 seconds, 15 seconds to answer that question. Okay, so let's look at those. Segment 1 would not code for messenger RNA. Both of them are DNA, so T is supposed to be there, okay? Segment 1 would be more soluble in water than segment 2 because it has more phosphate groups. No, each nucleotide has the same amount of phosphate groups. Segment 1 must be from a prokaryote because it has predominantly A's to T's. No. So segment 1 would become denatured at a lower temperature than would segment 2 because A to T base pairs have 2 hydrogen bonds, whereas G to C base pairs have 3. I hope that you got that answer, and C is right. Guided practice, we're going to walk through an FRQ and 2 little parts, well, maybe 4. There is an A, B, C, and D to this. And this is it's a cool question. In the box in the table, you see that there's a treatment 1, treatment 2, treatment 3, and treatment 4. And the medium that they're growing, these strains of yeast, okay, I can see that from looking at the top line of my reading. And we're going to have all amino acids present in the treatment 1, no amino acids present in treatment 2, all amino acids present except methionine in treatment 3, and all amino acids present except leucine in treatment 4. So let's look at our wild type, which is our normal strain, which does not have the mutations that they just put in this experiment. So the wild type can grow with it can reproduce with all amino acids present. It can reproduce with no amino acids present because it makes all of them then, okay? And then all amino acids present except methionine, yes, because it makes its own methionine. And all amino acids present except leucine, it still can make its own leucine. Now, mutant 1, let's take a look at that. Mutant 1 can it grow in all amino acids? Yes. Okay. Can it grow with no amino acids? No. That means that, and we'll read about it in a moment, that there's a mutation that they took out the ability to not produce something. Okay. So it can't grow without it. And then in treatment 3, it can't grow. All amino acids present except methionine. If it can't grow in this treatment, then it must not be able to produce methionine itself, right? Okay, in treatment 4, it can grow in all amino acids present except leucine. So I'm right here. And so if we can grow without the leucine, that means mutant 1 is making the leucine, okay? All right, mutant 2, we have all amino acids present. Yes, it can grow. No amino acids present. Nope, it can't grow because there's something it can't produce. And then it can grow in everything present except methionine. Yes, it makes its own methionine. And then everything present except leucine. No. So I hope you see something happening right here. Okay, now I'll read the text table. This table 1, the data shows that the growth of a haploid yeast strains on media that differ in amino acid content. A plus sign indicates that the yeast strains grow, and a negative sign indicates that the strains do not grow. Amino acid synthesis in yeast cells occurs through metabolic pathways, and enzymes in the synthesis pathways are encoded by different genes. The synthesis of a particular amino acid can be prevented by a mutation of a gene encoding an enzyme in the required pathway. A researcher conducted an experiment to determine the ability of yeast to grow on media that differ in amino acid content. Yeast can grow as both haploid and diploid cells. The researcher tested 2 different haploid yeast strains, mutant 1 and mutant 2, each of which has a single recessive mutation and a haploid wild strain. The resulting data are shown in table 1. So you have 2 things to do here. A, you're going to identify the role of treatment 1 in the experiment, okay? And then B, you're going to provide reasoning to explain how in mutant 1, okay, so mutant 1 can grow on treatment 1, but not grow on treatment 3. And I kind of gave you that answer a minute ago. All right, so I'm going to give you maybe 45 seconds to jot those down real quickly. Okay, about another 5 seconds. Okay, I'm not going to show you the answer yet because we have a C and a D. All right, well, let's see. All right, so a C. What did they do? They added a column to this table. Diploid cells produced by mating mutant 1 and mutant 2. So in C, yeast mate by fusing 2 haploid cells to make a diploid cell. In a second experiment, the researcher mates the 2 mutant haploid strains to produce diploid cells. Using the table provided, predict whether the diploid cells will grow on each of the 4 media. Use a plus sign to indicate growth and a minus sign to indicate no growth. So just write yourself a 1, 2, 3, 4, and then a plus or minus next to each of those. So you can do that quickly. I'll give you just a second. Okay. You ready? All right. So A. So the response identifies the role of treatment 1 as one of the following. And it says on the first one, positive control for yeast growth. And then the next one, to test the viability of all yeast strains. And then the third one, treatment 1 allows the researcher to be confident that changes in experimental outcome are due to differences. I've been thinking about this today. I've been thinking about this all day. I want you to say all 3 of these things when you're writing about a control. I want you to say it's the control. I want you to say that it's making sure everything else will live. And then I want you to also, no matter what the experiment is, to say to see if the if the results were due to the treatment, okay? You need to say all 3 of those things to just make sure that you have the right answer that'll be looked for when that question comes up. All right, and then B, the response explains that mutant 1 can use methionine when it is present in the medium, but mutant 1 cannot synthesize methionine. Okay, and then C. All right, I hope that you have a plus in every column because if all amino acids present, they definitely could grow. And no amino acids present, they can grow because why? Because one of them is producing leucine and one of them is producing methionine. So together as a diploid, they're able to produce everything in every situation. All right. Debrief and summary, our takeaways for today. The elements that make up each macromolecule, okay, you need to know those. The properties of the molecule determine its relationship with other molecules. The structure of the molecule is related to its function. You should also be able to describe the properties of the monomers and the types of bonds that connect the monomers in biological macromolecules. Explain how a change in the subunits of a polymer may lead to changes in structure or function of the macromolecules. Describe the structural similarities and differences between DNA and RNA, and predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. So I want you to describe it, explain it, and predict what you would do without it, okay, in almost everything that you do to prepare for this exam. All right, so common errors and misunderstandings. This isn't really a common error and misunderstanding, but it's a problem. And so I'll just read this to you. And I'm talking to my kids as well as you. This unit is the fundamental foundation for units to follow. Remembering the basic knowledge of the details in this unit will allow you to build your analytical skills as we move forward and then enable you to perform the skills needed to do well on the exam day. You will not have time to look up these facts and general knowledge about molecules. I recommend you develop online quizzes with your friends for the basic knowledge covered in Unit 1. You are not going to have time to go look this kind of stuff up even if you can have your notes. when you're to have 45 minutes to do everything that you need to do. All right. And then our homework for tonight, which is due tomorrow on Ms. Kate is going to talk about organelles and cell size. We're going to start Unit 2 in the next lesson. All right, so here's your homework. The stems and fruits of pineapple plants contain a group of protein digesting enzymes collectively called bromelain, and often used as an anti-browning agent for fruits and vegetables. Fruits and vegetables brown when they are bruised during transport or sliced and exposed to air. This browning is controlled by enzymatic pathways that produce brown pigments. The browning of fruits and vegetables reduces the nutritional value of the food, so anti-browning agents such as bromelain are used. All right, A, identify the type of monomer in which this enzyme is composed. That should be easy breezy. B, bromelain works by breaking the enzymes that cause browning into smaller molecules. Explain how the reaction that breaks up the enzymes occurs. That's like the second thing we talked about. A is the first thing we talked about. B is the second. And the C, we haven't talked about it, but I think that you're going to get this anyway. The pH of a solution determines the charge of certain R groups. The pH of pineapple fruit ranges from 3.5 to 5.2. Predict the effect on the activity of the bromelain if it is used in a product with a pH of 11, okay? And then provide reasoning to justify your prediction, okay? All right, so let's see, that's your homework. Device and internet access, do this by the 24th. We know that not all students have access to the internet or a device. We're working on student solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know, cb.org/tech. Please get that done by April 24th. And thanks for watching. I hope you'll join us again for the next lesson, for the beginning of Unit 2. Be sure that you do your homework question, and Ms. Kate will start that off tomorrow. Bye.